impacting all areas, teaching, extension programs, etc. So we try to promote education in AI, promote ethical issues, promote the use of AI, etc. So, and again, I'm repeating that myself, it's essential that we educate our students and cultivate a critical mindset, a critical thinking. So early in January this year, the rector approached me to write a project for the whole university uh, for one of the agencies, and which it was called, the project already submitted, the Multidisciplinary Artificial Intelligence Center for Research and Development of Advanced Application. And we, again, if you notice, the picture is very much similar to the picture of, we have the document of the Brazilian Academy of Science, and it was done in purpose because we follow the guidelines and the recommendations of that document. And so the idea is we have to shake UFRJ, create a multidisciplinary center, so exactly talking about many areas. And I just want to make sure in the beginning, it's not a project for someone or for myself or a group or for a group of AI or something or copy or any of the group. It's for the whole university. That's a key difference. It's the whole, it doesn't belong to anyone. In fact, the colleagues that help to with the project, I say, do you want to work a lot and not being rewarded at all, except having something that's good for the university. I said, okay, if you want to do that, let's talk. And everyone, hopefully, if the project gets accepted, will benefit. And also, we do not want to compete with other initiatives at all. So that was the we want. There are many initiatives in the university, individual initiatives, and projects. That's not the purpose. The purpose is have something that can benefit, especially promote research and benefit, hopefully, students and training human resource and for the productive sector. And collaboration is the key thing. And we exemplify as collaboration for national and international partners. And that's just a picture that I'm a very good artist, so I did with AI didn't quite went to how I like it, but it gives the idea. It has 4 pillars, the idea of the project. It's multidisciplinary, so we promote that, and we are not invented anything new. This kind of thing, it's happened. I say Columbia University has very similar ideas in the center. I have a colleague there that's advising the rector. In fact, Daniel knows him pretty well. And other universities in the world are doing pretty much similar thing. And we promote tools and infrastructure for research and thinking. So those who cannot afford having expensive machine, and I say I'm not competing with, we don't try to compete with LNCC that aims at a huge, big structure, or Lobo Carneiro that Alva is here and has a big center in supercomputing. The idea is very different. So we have machines that is relatively cheap, and we are calling Tier 4. I would say that Lobo Carneiro may be Tier 2. Yes, so we envision that at Tier 4. RMP is our research and education network that connects all the universities. They are pushing to a structure what they call Tier 3. So we envision that to be Tier 4. So something that is beneficial to many research, that they don't need very, very high-speed computer, and it promotes the use of the technology and teaching and access to technology. So Vision is two for the infrastructure for research and teaching, integrating with business sector, and AI education and ethical considerations that we need to do that. That's the infrastructure, but I don't have time to talk about that. Again, it's tier 4 that involved. And we have some multidisciplinary research that to exemplify throughout the university. So there are people contributing from biophysics and health. The health faculty of medicine is involved in that. And they work, let's say, faculty of medicine is working in geophysics? No. It's on other departments, statistics, one of them, and they work with statistics people. And because in biophysics, let's say, identify molecular interaction, I guess you're probably going to hear it from someone who is Aspucci is here. No? Okay. People from his lab. Okay. So they contribute these sentences from their research. So there will be other AI-assisted analysis of images that Senabio, it has a big equipment, and they also use LNCC premise, and they are contributing, and many other, let's say the medical school AI time series models, and they are working with different groups in the university. So they give collected examples of research, but anyone could use the infrastructure. It's or should be open to everyone. Material physics, so to develop predictive models on material properties, so the physics department involved. Engineering, computer science, of course, I am from computer science. And then theory, we try to put together projects on theory and also applications. There are from fine-tuning open source LLM, and again, we are focused on open source kind of thing, to be able to open source tools and models to be able to be used for everyone. And also focus on international cooperation. I won't have time to talk about the national cooperation, but we have a cooperation from INCT, which is, I don't know if a group of friends knows what it is, but it's a national projects that involve a bunch of people in Brazil, so are cooperating that in AI and international cooperation, so part of a big project with MIT, etc. And then for students, how many students you have here? Okay, several students. So I hope we get to you, and I claim I think it's nice, and get involved from now. So we need students from every center to try to put up something like that. So say you get people or faculty that will be willing to contribute, let's say like Daniel is here, and volunteer to mentor some students and then spread for all the universities. So you promoting, I was talking to the Macaés Center, and that's the same thing. Who wants to participate? So some students, okay, you're going to work for nothing. But knowledge would be the driving force, and get this integrate connection with the students and to promote the use of the education. In fact, we did pretty much that with CEDERJ. It's a distance learning initiative that started in 2005. Yeah, 2005, if I'm not mistaken, and I've been involved with it since 1999. And we didn't have students at that time in Europe. We didn't have a lot of them yet. So this is inspired in CEDERJ, that we had a series of students who helped spread the word to the interior of the States. And so we hope we replicate that, and we hope that we can get fellowships. So there are private sector that are contacted for fellowships. So we hope we can get that as well. So that's it. Thank you. Many things that I'd like to talk, but I hope at least I gave an idea. Thank you very much.
Muito obrigada. Thank you. Tem algumas perguntas? Question from our. We are also beside the French partner. We also have possibly a new partner, the Polytechnique school from Milan. We are here to representative. Thank you very much.
Oh, from Milan?
Milan, yes.
So, Torino has a similar center, if I recall. So it would be great that we have friends from Milan also. Yes, we are. Yes, I imagine. Right. And so it'd be great to. I was talking to a colleague that Daniel knows, Jim Burrows from UMass, also to cooperate in that. And hopefully we will get also with Sorbonne. They have a Senatente, and Purdue at the United States already gave a letter of. To be perfect, like I said, open AI, and everything free. That's what we aim for. Thank you.
More question? We have a coffee break,
Yes.
but you will be gone soon.
Okay.
Okay. Yes.
But
More questions?
she's volunteered already. She doesn't know, but
Okay. Next, we go to Daniel Sadat. That is from here, basically. He's from computer science. Daniel Sadat, for who doesn't know Daniel, is a very cooperating and very smart, intelligent person. He's part of the Academy of Science, and he's a person that is connecting everyone. So it's
Thanks a lot for the introduction, Stefanella. I know Stefanella since I was a master's student when Stefanella was doing her postdoc here.
Yes.
So
It's a lot.
I'm glad to be introduced by you, Stefanella. Thank you. So I don't know. I think, okay, maybe I should download it. Okay, so I will be very brief because I know we are short in time. But thanks, Stefanella, for the invitation. We are welcome colleagues from France and from Italy. We have collaborations across the globe here in UFRJ, so it's really a pleasure to receive you in person. And thank you all. So I'll be very brief. So my original field is computer networks, and now together with Edmundo and other colleagues, many of us are getting involved with artificial intelligence, machine learning. So I'm trying here to point some of the initiatives in which we were involved, bridging networks and artificial intelligence. So the first one talks about networks to support artificial intelligence. The second one talks about network concepts that inspired some ideas in artificial intelligence. And the third is the other way around, artificial intelligence for computer networks. Okay, so in the first one, we have here this reference on early exit with multi-arm bandits. So basically, we have these huge models, and it's a challenge how to actually deploy them. So one solution is to have early exits. So you run the model up to a certain point, and if you're satisfied with the answer, then you do an early exit and you say, okay, I'm fine with this. Otherwise, you go up to the last layer and then complete the task to see if you can get any additional advantage by doing so, right? So here the challenge is to reduce the inference time, right? And we use for this purpose multi-arm bandits. And basically, this kind of work is in the intersection between networks and artificial intelligence, right? Because we have a network, we have the cloud, we have the edge, we have our, let's say, devices. And basically, we have to decide what will run in the device, what will run in the edge, what we will send to the cloud, right? When will we do the early exit and not. And actually, this connection between artificial intelligence and networks goes even further, because we can now start thinking also about buffers. So people from networks love buffers, right? We love queues. And actually, we can relate queues and artificial intelligence, because these artificial intelligence models, they are fed by inputs that can be stored in buffers, right? So we can talk about delay, we can talk about losses, we can talk about all these cool concepts that people from networks are used to, but now also relating to artificial intelligence, okay? So that's one sort of work that we are involved with. Actually, this particular work is being presented this week, actually maybe today or tomorrow, at SBRC, which is a conference, Brazilian conference on networks. So colleagues here from UFRJ, some of them are not here because they are there presenting this work, okay? In Natal, it's in the north of Brazil. Okay, and now the second kind of work that I wanted to talk about is a work that is inspired by networks. So it's artificial intelligence inspired by networks. So as I told, my original field is networks. So Edmundo was my master's advisor, and also I was co-advisor for PhD and advisor for the whole life, right? So during this network class, one thing we learned is about Bloom filters, right? So actually, in this work here, we relate Bloom filters to another field, which is weightless neural networks. So there is an expert here at UFRJ, Professor Priscilla and Felipe França. They are experts in weightless neural networks. They have many works in this field. So basically, we noticed that actually these weightless neural networks, they can be seen as, let's say, generalizations of Bloom filters. Okay, so this is the contribution here. So we got an idea from computer networks, right? And use it to better understand an artificial intelligence tool, which is weightless neural networks. So here we have a picture that actually Priscilla shows in many of her presentations about weightless neural networks. And basically, when I saw this picture, I said, wow, this looks a lot like a Bloom filter. So basically, in a Bloom filter, we have these memories to the right. And then when you get an input, this input field feeds these memories, and then it sets some bits to 1. And then when you want to check if a pattern is related to that one, you see how many bits that are set to 1 also correspond to bits from the alternative pattern that would also be set to 1. So basically, this was the connection between weightless neural networks and Bloom filters. So yeah, we have a work on this topic as well. And then this third work here relates to fairness. So fairness is a very cool, very hot topic in artificial intelligence, right? But fairness has been studied for a long time by computer networking people, right? So in Jim Kuro's book, so Edmundo mentioned, so we have a strong collaboration with UMass. I did my PhD at UMass in big part because Edmundo introduced me to Don Todd and Jim Kuro's when I was an undergrad. We took a class from UMass online. This was very non-conventional at that time. And we learned about fairness, right? How to fairly allocate bandwidths in a network, right? So in this work, we try to get inspiration from these ideas to understand how to fairly allocate resources in artificial intelligence models, right? So basically, this is an example of this sort of application. So here we have a news feed. Actually, this was collected, people from Milan, maybe you'll be familiar with some of, so for instance, there is a M5S here, which is a political party in Italy. Actually, Stefanella also. So it's a movement 5-star. So basically, we collected data from the Italian elections, and then we tried to see how the different artificial intelligence bots were allocating the resources, which in this case are like the space in the timelines of the users, based on their orientation. So we see, for instance, that the undecided bot is not let's say, fairly splitting its attention between all sorts of political orientations, right? So basically, yeah, we can see these machine learning tools and these artificial intelligence models as also, let's say, tools that allocate resources across different streams. And this is exactly the kind of problem that people from computer networks have been studying for a long time, which is how to allocate resources across users. So yeah, this is another kind of work in this direction. And finally, this is the last one, I think. So I had to pick some specific works, but this one, for instance, is involved with Kadu also, that is here. It is a collaboration with colleagues from Siemens, where we have been using artificial intelligence and other techniques for cybersecurity, right? So to understand the National Vulnerability Database, to understand some severity metrics like the CVSS metric. So so basically here we try to tune these models to specific products. So the input is the NVD text and the output is the scores. So the takeaways here are that we have that networks may help to make artificial intelligence more efficient, right? Network structures and goals can inspire artificial intelligence, and the other way around as well, right? That artificial intelligence can enhance networks. So I just wanted to point these connections between artificial intelligence and networks. Yeah, thank you very much.
Some question for Daniel? Question?
I have one point.
Yes.
Hi. Can you give me just an impression of the advantage that I can have if I use the intelligence, the AI for networks? I mean, is it more related to speed of the network that will be improved or whatever?
Yeah. So basically, network is a very broad field, right? So we have many infrastructures that are already over provisioned. So for them, we don't need to do much. But actually, in many situations, we do need to add intelligence. So to reduce costs, for instance. So just to give an example, how to set the contents in the caches, right? So we have caches in the service providers.
Okay.
Netflix, for instance, distributes caches all over the world and needs to decide which content to put in the caches. So this is an example where artificial intelligence can help, but there are many other examples.
Makes sense.
Yeah.
Thanks.
Thank you.
So next, we are in the run. Álvaro Coutinho from Copy and from civil engineering. And he has a lab, a multidisciplinary lab, and also he will be in charge of the new center. Yeah. He will talk about this very new adventure of UFRJ.
First of all, personal touch. This is my son. He graduated in the Polytechnique in 2007, and with me, with my very good friend Patrick. At that time, I think he was one of the well, I was a professor, and then I think at that time he was one of the position directors in the Polytechnique. And that lady here that I have forgot her name. Elizabeth. He was a chef in the
She's my former boss.
Oh, okay. And then so it's a very personal touch and a welcome. Getting down to business. Yeah, what I am talking today is the Center for Excellence in Artificial Intelligence for Renewable Energy. I am the PI, and I have my email here. Well, this is one of the national centers. It's a competitive project that we won, and is led by a COPY. COPY stands for the Graduate Center for Research in Engineering at UFRJ, where Edmundo and I and some of the other colleagues here are affiliated. And we have all those partners here in science and technology. We have these international partners in France, India, and in Spain, Barcelona supercomputer centers. We have some companies involved as well, founding companies and non-founding companies. And we have several innovation initiatives around us and some projects in many companies in the energy sector in Brazil. The center organization is more or less like that. And on the technical aspect, we have working groups in wind, solar, biogas, hydrogen, and transversal working group that provides, input in AI, especially machine learning, HPC, and data science, because the level of use in these different different industries or applications is very different. Okay, and then we need to have this transversal working group. This is the leadership. I am the PI. We have people from NNCC and several people from UFRJ here. It's interesting to see. I also have an academic position, so I'm the head of interdisciplinary area of computational engineering and science at COPY. But we have people from applied math, optimization, computer science, mechanical engineering, energy, chemical engineering, and safety energy. Okay. The location of the center is at that building in our science park, our technological park. This is where we have our advanced computing facilities, visualization facilities, and remote sense facilities. Actually, the center is one room, and it's distributed all over the university. As a bit of context, if you allow me to say, my point here is that this is from Google Ngram Viewer, and I did a search on AI, machine learning, HPC, and computational science and computational mechanics on this. We see clearly the initial hype on AI, and then everything, the trends is going up. And this here somehow is hidden, which means basically the necessary infrastructure to do AI. And I would not say this AI is no longer pure computer science. I would say computational engineering, computational mechanics, which more or less encompasses all fields of application of interest nowadays in AI. More or less, let's say. The convergency in infrastructure have already occurred. I quote this to machines here is more important for you guys, for everybody to understand that those machines are huge, and even a machine that do transaction in social network like Google, and a machine dedicated to high-level computations, they have more or less the same architecture. You see, this is already happening, and in Brazil we see this happening as well. We have in Campinas a machine on a private center, a research center in Campinas City, which is funded by Samsung Brazil, which is a machine already in place for AI, and the biggest machine for open source, open science here in Brazil, which is located in Petrópolis in this state. So more or less we have the same situation. Now, if you allow me, I will walk you through briefly in jumping in some of our main, some of my, now I'm speaking not as the leader of the center, but the leader of my smaller group. So basically, concerned with data-driven reduced learning model for solving large-scale problems, like some of those found in geosciences or engineering systems, more or less. This has been computed in our supercomputer here at UFRJ. And the idea here is this nice solution of this couple multi-physics problem take a long of time. And the idea here is to use AI or in order to produce surrogates models for allowing more expedited exploration of parametric space, which is a fundamental thing in engineering and natural sciences. So I mean, we are addressing here in this question here in red, which more or less say what we do if we even compress floating points. More or less we have here is this workflow. We have an offline phase in a big machine, and then we have a online phase in your workstation or laptop. This takes hours and or even days. This is a matter of seconds. And if we take a look on this, we can do a very good job in compressing the data. Of course, I'm saying that it'd be interesting to look at single value, see that compresses the data to the point that mess around with the things, so we can have a very nice reconstruction of the PD for this complex multiphysics problem. Okay, jumping again for some sort of flow assurance. Here we have a bubble, and the idea here is we'll jump to this, and it's in order. Well, I mean, usually neural networks don't know anything but the data about the problem at hand. So how to inform a neural network about the physics? This is what we call physics-aware, physics-informed machine learning. We extend the standard losses by imposing a certain mathematical model that we all agree that's interesting for the problem at hand. Here is the Navier-Stokes equation coupled with Eichhorn equation for simulating multi-phase flow. And then outside the data, we rely on this to complement in enforcing this regularization in the neural networks. And of course, we are able then, by knowing only the bubble position, infer all other fields of interest. Okay, this is what we got. Go ahead. Well, neural networks are very good things in the sense that approximate very well functions. But what about operators? We are working now with neural operators with our extensions of all this, capable of learning operator between direct spaces. So we are working with these neural operators for a forward forming version with a problem for interested geosciences. Well, one of those things is called a depot net that combines two networks. And with this, we can produce a very good approximation for this very interesting, very difficult wave propagation problems. We can reproduce time series as well as the spectra, the time spectra very, very well with this technology. Ambition, of course, everything. I have some statistics here. Everything needs error bars. So we're doing a lot of uncertain quantification, particularly in seismic imaging. And this is more or less the standard stuff on top. And this is when everything is probabilistic for seismic imaging. And believe me, it's very difficult to convince energy companies that they have to move our incorporation towards incorporation uncertainties, because the computational complexity is much more demanding in this case. But thanks to machine learning, we are able to do this. Just at a glance, take a look at this phase of the workflow. If we do by brute force, well, you can run everything and cut, let's say, 1/3 of the processing time. But is it enough? No. We have to rely a lot on deep learning in order to provide surrogates for all those components in this geoscience workflow. And of course, we did that, and then we are getting more and more results. As a final remarks, everything I'm talking here to you today is in the emerging field, what we call scientific machine learning, which is a branch of computational science in the intersection of machine learning and scientific computing. This physics-aware machine learning can be applied to a broad range of relevant problems in engineering and science, and we have shown advances in 4 specific cases: reduced order model, physically formed neural networks, neural operator, and uncertain quantification. And we foresee that the impact of this scientific machine learning in all aspects of engineering and science will grow enormously in the coming years, and we are open for collaborations. This is the team. Probably recognize some of those people around here, and thank you for attention.
Some question for Aldo?
I have 2 questions regarding the seismic applications, if you may. So the first one is on the DeepNet for the operating version of FWI. How stable is it? Because I would assume that if you change geology, it'd be very finicky. Because FWI is fairly delicate.
I know.
When you change geology.
Of course, if you pick up initial configurations, good initial configuration, everything goes well.
Yeah.
Okay, but sometimes it's hard, and then you have to run pre-computations in order to get a good idea, and then.
But you shouldn't be able, for example, to do a DeepNet training for one size geology and transpose this to another.
Neural operator is meant to do this.
It's meant, but is able to.
Is able to at a certain point. The variations cannot, it's still, but that much depends on the amount of data you have to train. That requires a huge amount of data.
Huge or obscene?
No, huge.
Okay. Second question: have you tried to?
In my experience, but come on again. We never run a full 3D problem in this scale.
Okay. You've never tried?
No, not yet.
Well, for example, for a Lula field or something like that.
No, but I forgot to say we are doing this in a Bayesian perspective.
Okay. Last question: have you ever tried? Probably not, but have you tried to do like an angle getter, RTM uncertain quantification or something like that? Offset dependent RTM uncertain quantification, because it becomes much more noisier.
Yeah,
RTM.
We did some experiences with noisy fields, increasing the noisy level.
Probably becomes a nightmare.
Yeah, but again, look. Well, thanks God, not everything is solved. Right?
Yeah, for sure.
But those are point towards a game changing in the way we see those problems.
Yeah, I remember. Yeah.
All right. And
thank you very much, Aldo. So the next speaker is from the Institut Mathematics. It's Fabio Ramos. Thank you, Fabio.
Thanks, Stefanella, for the kind invitation. Well, my name is Fabio. I'll be very brief. We're hungry. I need some coffee. Okay. So I'll just some slides, right? So I beg your pardon. I'm actually a pure mathematician, and at some point I realized that 90% of its works of real is oil and gas. So people are killing themselves for some very little grant. And I'm sorry, there's just a lot of money to do your research in other directions. So you better go in. So I can't keep myself from asking fundamental questions. So yes, we have some results that are completely theoretical. So Just one, so I'm just talking about the results from this year, right? So this is work where we use neural networks to find similarity in physics, physical laws, right? I do not get into a lot of details, but we essentially exploit scaling and symmetry laws. There are a lot of symmetry groups hidden in physics. So the core idea is to use this neural network we call BarenNet in honor of the great mathematician Barenboim, right? So the BarenNet and also is an acronym for BareNet. So automatically identifies similarity relations from data, right? And so that's an application for non-Newtonian fluid mechanics. So we can, let's say, collapse curves. That's what we can do. Give me a lot of curves, or maybe with a lot of parameters, and I just can collapse them by finding out hidden symmetry groups. Right? So this is the outcome of this work. This is actually a PhD thesis from my student. So there's the design of neural network, just to give an impression, but doesn't mean anything right now. Right? So yeah, so we are also asking ourselves about fundamental questions. Actually, that's what drives most of my group interest in neural networks, people are interested in fundamental questions. But their actual scholarships come from oil and gas because you just the scholarships are 10 times higher. So there's nothing you can do. But yes, we have applications. We enjoy doing the application, right? We really enjoy delivering the product. But the kids actually, they really like the that's myself. So this is a collaboration with ExxonMobil. That's a project that has two fronts, geoscience and oceanography, which is weird. Our project can attack geoscience and oceanography at the same time, right? That's because we use these diffusion models. The diffusion models is what you use when you ask ChatGPT to create an image. So they are actually running a diffusion model there. The objective in geoscience is to generate high-resolution core-scale 3D digital rock and translate between modalities of images. And in oceanography, ocean sciences, we want to predict fast ocean currents at a given point of interest. And we're using essentially the same technique, which is here, right? Okay, so somehow what we are building something like this, you get CT images, you get images from FID-SEM, some descriptive characteristics, and then we build a lot of core-scale generation to do uncertainty quantification, right? And then this is working already, right? This is a picture of my Chihuahua, Jimmy, and how we learn this stuff. What I mean by learning, how can I say that I really know Jimmy? It's because even when he's completely noisy, I can recognize him, right? You can just put a lot of noise and I can still look at him and like, okay, that's Jimmy, right? There's nothing you can do, but just see an ear as it is Jimmy, right? And learning is actually this double process of encoding and generating. So you actually generate a lot of noise, and then in my brain, I automatically denoise and recognize you, right? So this is the idea of diffusion model. You can do this for probability distribution. You can get a probability distribution with a lot of nodes, and I add a lot of noise, and the probability distribution becomes a Gaussian distribution, right? And then I learn to noise the probability distribution and to denoise from Gaussian distribution to the original one. So if I can do this, I learn the data set. That's the kind of thing we are trying to do. These people are very attractive, but they're all fake. They were built by using this training data, the celebrity, high quality data set. So what these guys did in 2020 is exactly this. They get like a picture of Tom Cruise, and then they add a lot of noise, and then denoise. And then Angelina Jolie, noise and denoise. Tiger Woods, noise and denoise. They do this a lot. 30,000 times. And then they learn to go from pure noise to some pretty people, right? And then I learned this process, and then you learn how to, from pure noise, you just randomly generate pure noise, and then you try to get rid of the noise, and then you end up at some face that looks like a real face and attractable. So that's the sort of thing diffusion model does. So we are doing this for rocks, for digital rocks, right? So we add noise, take noise back, and there are a lot of complications there. I'm not going into the details, but that's what you're doing, right? And the outcome of this, so this project started like a year and a half, and we already have some results, probably published very soon. But we have codes. So it's open and it's nice, right? So it's open science. It's funded by Action, but everything is published, it's open, the code is open, you can just go there and use it, right? We have two packages. One is for generating rocks, and the DiffSci is a general framework to use diffusion models for scientific purposes, right? The second front is to use these diffusion models to do forecasts of high-speed currents. I don't have time to talk about this. It's way more complicated because it's a dynamical problem, so you have to generate dynamical systems, and it's way more difficult than I thought, but it's fun. All right, have some results. I'm going to talk about this. We also have some other projects with Galp, is another oil company, to use like GPU accelerated methods for running flow assurance simulations. We had a project with Shell to predict the growth of calcium carbonate in the pre-salt, and also have all the other projects. And that's it. Right? We have all this sort of fun stuff going on, and it would be an honor to collaborate with you. Thank you.
Thank you, Fabio. Thank you. So I guess, there are people from EGO? Elizabeth? No? No? Yeah. So, Monica? Maybe some people got lost. So we just, before the coffee break, we just start with one from we start the next section because I don't know where the people from geophysics, we lost them. So we start with the group of people from application to biomedical application.
Okay. Yes.
So Pedro Torres.
From biophysics.
Institut of Biochemistry.
Biophysics.
And?
Biophysics.
I'm sorry, biophysics. We have Institute of Biophysics, Biochemistry, and Biochemistry and Medical Application. No?
Yes. Okay. So let me use the microphone to. Okay. So good afternoon, everyone. I'll try to be brief. Thank you for the invitation. Originally I didn't know that this talk would have to be spoken in English, so I tried to adapt as much as I could the slides. So I'm going to talk about machine learning in the identification of novel therapeutic agents for emerging diseases. I'm going to start
by telling you about the problem we're trying to face here. I mean, in the lab, we try to deal with many different problems regarding molecular interactions and diseases in general. But today I'm going to try and talk about a particular project that involves machine learning. So this is a timeline of my antimicrobial resistance, the emergence of my antimicrobial resistance throughout history. So this here shows that penicillin that was discovered in 1940s, less than 10 years after it was discovered, in the bottom bit here of this timeline, we can see that the bacteria and microorganisms, they developed resistance for these particular drug. And this happens for every single antibiotic we have discovered so far. This is a foreseeable problem. So this is an excerpt from Sir Alexander Fleming, Nobel lecturer in 1945. He said that the time may come when the penicillin can be bought by anyone in the shops. Then there's danger that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant. So this has been predicted since, well, the dawn of antimicrobial use. So there is some prediction that indicates that in 10 years, or I mean in 20 years, maybe 25 years, the antimicrobial resistance related deaths will surpass the amount of deaths that we have today from cancer. So imagine, people from your, I mean, everyone has familiar people that have had cancer in their lives. Imagine that in 25 years, this will be true for people that have been infected with resistant bacteria. So why are they so hard to kill, these bacteria? Because, I mean, there are some targets, specific targets that these antimicrobial molecules, they target. And these targets, they have been used many times throughout history, and they have some characteristics. So this study from 2006, they started with bacteria, and they had 2,200 proteins with metabolic function, with known metabolic function. 155 of them were predicted as promising targets. From these 155 proteins, we had 64 targets that were conserved among the pathogens, the bacteria. And from these, many of them were already known. So from 2,200 proteins, we ended up selecting the same targets that we use today in clinic. So this is a study in Nature Biotechnology, 2006. So good targets, I mean, to kill bacteria, we have to find targets that have high essentiality. It means that they are part of multiple metabolic pathways, I mean, for bacteria. They have low degeneracy, which means when you inhibit that target, then you kill the bacteria because it has no other proteins capable of doing that same job. They must be highly druggable. So this means that the target has to be inhibited by small molecules and drugs. And it must have well-defined pockets, binding pockets. It must be bioaccessible. Extracellular targets usually are more bioaccessible than intracellular targets. And extracellular pathogens are usually more bioaccessible than intracellular pathogens. Low similarity to host proteins, meaning that when we inhibit bacterial proteins, we don't want to also inhibit human or host proteins. And low mutability, meaning that this protein shouldn't be able to just mutate and evade the treatment. So all of these are characteristics of a good target. And the bottom line here is that good targets are hard to come by because they are weak spots. And they are negatively selected throughout evolution. With this in mind, I mean, this huge problem, the idea was how can we lever computer power to actually try to tackle this problem? And I came up with this idea of a perpetual machine for drug prospection, for drug candidates prospection, and it should have like a cycle of continuous prospection, where three pillars are actually tackled. These three pillars would be targets prioritization, and this would come from metabolic network modeling. So we get the bacteria, we know all its genes, we create a metabolic network, and we pinpoint genes that should be essential to kill that bacteria. Then we come up with the structure of these molecules. So there are many different ways, including machine learning based ways. Probably some of you have already heard about AlphaFold, which is a software to actually predict the structure of proteins, which has been a problem in science since forever. So we can model the proteome of these bacteria. We prioritize the targets, then we start looking for molecules that are able to inhibit these targets. And this is done by virtual screening. So we're going to virtually screen a huge library of molecules, like billions of molecules. This will come from a ligand database, and this ligand database will be pre-filtered, and then these molecules will try to bind to the targets we have selected. Then we use machine learning again to perform a consensus scoring of the interaction between ligands and targets. And then we feed a database of putative, of hypothetical target and molecule complexes. Okay? So this should, in theory, speed up the process of finding new antibiotics, and this will be organism specific. So we'll have one of these machines trained to deal with Klebsiella pneumoniae, one of these machines trained to deal with Mycobacterium tuberculosis, one of these machines trained to deal with Mycobacterium leprae. So the idea is to have many of these machines, one of each, trained to deal with one specific pathogen at a time. And then this will be kept like forever, running 24/7. Then scientists can come up in this database, select a few molecules that they want to test in the lab, and then test the molecules with higher probability of actually finding a new drug. So the modules are TARDIS. Then I'm going to speed up a little because I think we have like 10 minutes only. Probably it's already 7 or something like this. So TARDIS is the targets discoverer. This will build up a network of metabolic interactions. This is done by getting the annotated genome of the pathogen. We have specific methods to build these metabolic networks. They are called the restraint-based methods. Then we build the metabolic networks. We pinpoint the weak spots. The weak spots might be, they might be called the choke points in the metabolism. Choke points are points where you actually have one single path between one substance and other substance in the path. So then we can, inhibiting that, we can disturb a lot the metabolic network across. Then this is the student Camila. She has built a few of these metabolic networks, and she has come up with in Cambridge, and this is a protein complexes and homo-oligomerics interfaces resolver. So this builds models, and this was pre-AlphaFold. So when AlphaFold came up, it basically just like destroyed everything I had done in my previous postdoc because it just surpassed everything in terms of quality but we're still trying to use this, and then in cases where this software don't deliver, then we try and use AlphaFold. AlphaFold has its problems. So this is the actual pipeline. It uses several tools that are already established in the literature, in the biochemical literature, the structural biochemical literature. I'm not going to go through this module. Then there's SPADA, which is a module to actually minimize and optimize these proteins, so they are not like crystal structures anymore. They are structures that are more likely to be found in natura, so in vivo or in solution. They are just, let's put it like this, they are less artificial structures. Then this is the module that's going to do that. This is the student, Guilherme, which is working on this module. So this module involves putting the protein structure that we have got from the previous modules in a water box and simulating this a little to just relax the system and so on and so forth. Then Hocus Pockets is a module where we're going to find the specific parts of the protein where we can try and bind the ligand. So this is being done by Luisa, and she's using several different software to find these cavities in the surface of the protein. Some of these programs also use virtual intelligence, artificial intelligence. So we come up with the pockets, which are spots in the protein surface where we can try and bind our putative drugs, our candidate drugs. And then Librezil is another module which is also being developed by Guilherme, which tries to cluster up all billions of molecules that come from the ligand databases. So these are chemical structure of small molecules that are going to be docked into the protein pockets. So this is a module where we're going to try and reduce the number of actual molecules we're going to try and bind by clustering them up. And this is also going to be done with artificial intelligence, particularly with non-supervised methods. And then finally, OC Docker, which is the Open Consensus Docker, which tries and gets the results of the docking, which is the process of actually binding the target with the small molecule, and it tries and comes up with a consensus among various different docking software. The three of them are here: Vina, Smina, and Plants. These are three different docking softwares. They are going to provide scores, and we are going to take these scores alongside many other properties, both from the ligand and from the receptor, and feed these to supervise machine learning models. And this is already being done, and this is the result. So these lots of points here, they represent the consensus scores that have been generated by machine learning methods, and these are the ones generated from classical methods such as average or voting schemes and so on and so forth. So we can see that machine learning can actually enhance a lot our area under the curve, which is the AUC, to actually measure the power of our distinction between ligands and non-ligands. Next steps would be to integrate these methods in a continuous pipeline. The individual validation of the methodology, so each one of these modules will have to be validated. The methodology as a whole, the whole pipeline, should also be validated. And this will be done, of course, with in parallel with wet lab collaborators. And this is the last step would be the construction of this pipeline for microorganisms of interest like Pseudomonas aeruginosa, Klebsiella pneumoniae, Mycobacterium leprae, and so on and so forth. This is the escape group of bacteria that are considered dangerous bacteria by the Global Health Organization, World Health Organization. And well, this is the group, and I hope I didn't exceed me too much. Okay, thank you very much.
I have one question. Thank you. Pedro, how do you deal with this idea? I mean, you are preparing compounds that can inhibit an enzyme that is probably unique, or at least as unique as possible, regarding the enzyme that has it in humans. But how can you test whether this compound is actually specific for not only this enzyme, but all the other enzymes? I mean, you have to.
Yeah, sure.
Thank you.
So, of course, we're not going to be able to, I mean, we as a group to validate every single prediction, but our idea is to pinpoint some of these complexes, some of these small molecule target complexes, and then take it to the wet lab in terms of mainly ITC, like isothermal calorimetry, and some enzymatic assays. And of course, we're going to also try some in vivo assays with people from the Institute of Microbiology. You can have some collaborators there, Professor Raquel, yeah, in the Microbiology Institute.
Okay. Thank you.
Okay.
So one thing that I noted, I'm not from the life sciences, but your idea is to target always one small molecule for one critical path. And for me, it feels that you're inducing a wide rabbit chase, because you target one critical path, you do a negative selection, then you need to follow up.
Yep.
If instead you try to not get one critical path, but a plethora of mild paths that have the net effect of collapsing the bacteria, you have a more complex, maybe, but in effect different optimization problems. For the bacteria, you'd still need to induce evolution, but it would be a more complex evolution to move out of this minimum, because it's not one gene that you need to move. You need to move out many genes. It would be a more stable configuration, and maybe you would be able to have a cocktail of substances. How would you think about the situation in terms both of the from the biological point of view and from the computational point of view?
Well, that's a very, very good point. I mean, I hadn't envisioned this possibility here for AM to PM, which is the name we've given to the whole pipeline. So we hadn't envisioned this possibility, but I think that's precisely what happened with HIV.
Yeah, you
So,
have a
yeah.
cocktail drug.
Yeah, but that happened along decades of treatment and research, because many drugs were developed individually, and then at some point in the future, from when you look at the perspective of that time, these drugs were combined in a cocktail, and that made it more difficult for the virus to evolve and escape the treatment. But that wasn't envisioned a priori. Maybe we can try and think like this, but that would have to, so our regulatory agencies drug usually takes like 15 years to be developed. I don't know if 5 would take.
But you're developing as a
7 to
package.
5. Yeah, I know.
Yeah.
I understand the idea, and I think it's very interesting.
Because you're co-developing a package.
Yeah, I just don't know how practical it is, but I'm surely going to think about it.
Because this is a system, right? You have a in silico drug design package.
Yeah.
You can design for a drug or for a cocktail.
True.
It's just how you run the software, right?
True. Correct. Thank you for the suggestion. Look into that.
First of all, thanks for the presentation. I'd like to also say that as an undergrad student, I don't have any expertise yet, and if I had, it wouldn't definitely be biology. But I've heard that the real concern with the superbacteria are those that can arise from animals, like from the meat industry, that they use indiscriminately the the antibiotics, and then these superbugs can hop for humans. And I want to ask, how true is that assessment, and how can your project tackle that?
Well, do I need to? Thank you. Yeah. So that's precisely true. I mean, usually the problem with animals, we usually think as animals as reservoirs of actually viruses. But bacteria are also a problem because of, as you said, the meat industry, they use antibiotics like indiscriminately. So yes, that's precisely true. Drugs are becoming, bugs are becoming more and more resistant due to indiscriminate antibiotic use, including in the industry. How can this help? So I'm trying to do this for one species, one bacteria. But then, as you saw, we're trying to do this for Klebsiella, for mycobacteria, for Pseudomonas. Okay, so like start with 3. But we can try and make like huge clusters, computers, maybe virtual machines, Docker, Docker containers filled up with machines. Each one of these thousands of machines could be working for a different bug. And then we could have a huge database that could identify new possible molecules for all microorganisms that are important as a antimicrobial superbacteria. So we'll start with 1 to try and prove our point, and then if we see that it works, then we can scale this up and try to predict drugs for everything. So that's the idea. I have to start somewhere, but yeah, true.
Yeah, we are going to get a coffee break now. We need to take a coffee. We'll come back in 15 minutes, okay? So the speaker, well, we go to Cesariani because there we have bathroom working, and the coffee is waiting for us there. Okay, so all of us, because the foreign encounter.
basically, we are interested in my laboratory to understand metabolism, how cells and organisms transform energy from the environment to make them alive. And I'm particularly interested on a specific organelle inside the cells called mitochondria. Some of you may know it. That's the powerhouse of the cells, where the energy comes from the outside of the environment to transform energy inside the cells. So one of the things that I'm going to show briefly is how I'm applying AI, and specifically the AlphaFold algorithm to predict protein structure. And then a second part of my talk, I'm going to talk about the interventions that I'm applying AI in the classroom to improve teaching. Where it is? Okay. In the classroom. So next. Okay. So I'll briefly, I'm not an expert on AlphaFold. I have other colleagues here that are more familiar with, but as a user, we can now predict using this algorithm protein structure models to understand differences between particular domains and parts of proteins that are different between organisms in order to understand not only biology but also targets for drugs, for example, as Pedro has shown in his presentation. So we can, it's based on DeepMind, can predict 3D structures of DNA, ligand structures, and protein, and it's validated by multiple times using this algorithm. And so it's a very powerful tool. And as a user, as an external user, I can tell you that this is a very nice tool, and that we can apply not only in the lab, but also in the classroom too. So the problem that we are, the problem that we are trying to tackle in our laboratory is to understand how this tiny part of the cells that oxidize, that burns the food in our cells, the mitochondria, how this organelle works in terms of how the amount of energy that comes in is transformed into fuel, as ATP, which is the cellular fuel in our cells. And I'm particularly interested in a metabolic pathway that is the glycerophosphate shuttle. I'm not going to go through the details here, but it's a mechanism to transport electrons outside of the mitochondria to inside mitochondria to make them to produce ATP. It's a metabolic pathway that is not fully understood in karyotic models, but it's critical to allow ATP production, the fuel production in the cells. So one of the enzymes that participate in this metabolic pathway are known in mammalians, but I'm using a particular organism in trying to get a better understanding of this metabolic pathway, which is the Drosophila melanogaster fruit flies, which is one of the biggest and more powerful biological models to process forward. And using the Drosophila melanogaster, which has the whole genome sequence, transcriptome, and all the omics known in this organism model, we can, using the AlphaFold, we can generate protein structure models. This is just one of the proteins involved in that metabolic pathway, the glycerophosphate dehydrogenase, which is it's in golden here, and the Drosophila protein is this green grayish structure. So it's very similar. The fly protein is very similar to the human protein, and even the domains that are directly involved in the catalytic activity of this enzyme. So for this enzyme, we can say that this protein is very similar from fly to human. But we also have other examples that the proteins are not that similar, which is interesting to understand better biology. And then you can transform this scientific knowledge to discuss basic concepts in biochemistry in the classroom setup also.
This is another example, another enzyme of this metabolic pathway, the glycerophosphate oxidase. So all the domains, the classical domains of this enzyme are conserved between the two proteins. So it's a very powerful tool that we are trying to figure out the differences, the evolutionary differences between these two organisms, and to then because of that, then understand biology. So this is basically what I'm directly involved in terms of research using AI. But then I want to bring a second topic, which is, I think is the reason of why I came here, I was invited here, is to discuss how we can exploit the power of AI in the classroom setting. So, próximo, por favor. And everything for me changed after this day, when I figured out that ChatGPT came to the world, and then I tried to first of all understand what is going on, which this feeling I can tell you that I have the same feeling today. So we are overwhelmed by new tools day by day, and as a teacher, I was very worried since I was hired here to develop the critical thinking for students. And the way that I was doing up to the ChatGPT was to present the students the original data of papers, scientific papers, and then let them to figure out critically the original data of papers. But the way that I was doing up to 2022, I had to change, and I'm still trying to understand and applying that. But since then, I try to use and educate myself to use these tools, and then I learned that I can use them to let me know how to use them, which is nice. So it's kind of it's the feeling that I have is that it's a kind of if you chat with a very knowledgeable friend, and then you can ask them the very basic questions, and then he tells exactly what you need. And that's what happened to me with ChatGPT first, and then I became more familiar with other algorithms. So the activity that I developed, which I give classes of bioenergetics, so how the cells transform energy from the environment to the cell. So the bioenergetic, and then I developed this activity called bioenergetic AI, which is a kind of very nice acronym for that activity. So basically what I do in the classroom setting is to divide the class in five, 6 different groups. Every group chooses a topic, a relevant topic in biochemistry. Usually in a disease thinking, like cancer or diabetes or obesity, you name it. And then I ask them to generate a prompt, but a sound prompt, not a general or a superficial prompt, a designed prompt, using the knowledge that we discussed in the classrooms to ask ChatGPT first, and then I develop train them to use other algorithms, and then the ChatGPT answers. Sorry for the low resolution, but this was an example of a question in this setting, and then the chat answers the question. So the second part, this is the first part of the activity, the próximo, por favor. O próximo. And then the students had to search one of the key tools, online tools to find scientific papers, which is the PubMed. And then they go to the PubMed and check papers, original papers, that can support or refuse the aspect that ChatGPT had provided in the answer, using original papers. And then I give a class how to use the PubMed, which is not familiar, which is not easy for the young students to know to use it. And then we'll próximo, por favor. And then they had to prepare a short report, 5-page report, with with a critical analysis of the ChatGPT report, using the original data that they find in the papers, and then they discuss using the figures, using the data, the images, the tables, and after that they present this report for the class. And I can tell you, this activity is amazing. I did not design that. I asked consensus to prepare that, which is it was very nice. So the app is really clever to think on how we can use these tools as a teaching activity for classroom problem. So this is one example of the reports that the students came with the original figure of the papers, and then they discussed what the ChatGPT did good or did bad, and exactly why, and then we discussed that to use, and then they learned how valuable at least the ChatGPT had at that time. Now I can tell you that for biochemistry and metabolism, ChatGPT is more clever now than at that time. So it's learning, really. But I teach them all the tools too. So, and then they present that with a 25 minutes presentation, and then we discuss, we open. Usually the class that used to take 2 hours takes 4, 5 hours. I had to kick the students out of the room because they want to stay in the room because it's so nice, it's so engaging. And it's a very, very powerful tool. It really changed the way that I was giving lectures in this discipline. So we'll proceed forward. So that is the idea, is to show some of the applications, superficial applications in my research using AlphaFold that can be explored, but also, and finally, I just want to express some of my concerns, which is for the students that are really worried with the technology. I think the AI is a very nice tool because they can use as a way to understand and to develop critical thinking. But the problem is that for those that are not, that can cheat. And I think that is an ethical issue that all universities have to face. And I give an example. In my perspective, I think we have to change all the ways that exams are provided for the students everywhere. Second, I think that for graduate courses, I think the PhD defense as a document, as we always had, I think we had to discuss that, especially at the introduction section, which is very easy for the student to develop this kind of very nice and knowledgeable document. So I think these are open questions, but I think this is the ethical issue I think we have to face worldwide on that. Thank you.
Question?
Have you tried using ChatGPT or other of these services for do very intricate or very complex research, like deep search?
Yes.
What's your experience? Because for me, that's by far one of the best values that they brought for me.
The ChatGPT itself?
More DeepSeek, actually.
DeepSeek.
DeepSeek and Claude are better than ChatGPT most of the time, because I can tailor a very sophisticated prompt, and they can search on the web and filter through PubMed, filter through archive, and get me some references. I would take hours to find.
Yeah. Mm-hmm. So I can share my brief experience on that. I'm not an expert on AI, not at all, but I'm now educating myself on that. And for that, one of the ways that I'm doing is to get closer with friends that are really professional. That Moisés Marinho, he's one of the professors in our university that is leading a group here at NUTIS that are focusing on trying to understand the impact of AI on teaching and on education. From my perspective in biomedical area, ChatGPT it used to be bad, but it's learning, and now I'm worried in a way that the students can cheat. But DeepSeek, I think it's better for biomedical science. But I'm really impressed with Gemini Deep Research. This one,
That's
the
interesting.
Gemini Deep Research, I think this one is transforming. It's really, really good on that. And even the hallucinations that I use it to see that in the ChatGPT, I never see an hallucination like those in ChatGPT with Gemini Deep Research.
Yeah. So the problem of hallucination I see a lot when I try to produce text, like write a text for me. I don't have this problem when I say find me references about, and I try to get me stuff. I rarely have problems with hallucinations.
With ChatGPT.
Yeah, with either of them. I usually don't bother with ChatGPT nowadays. I only bother with like the O3 Pro or the O4, which are the better ones. I usually use either the DeepSeek V3, the R1, or the Pro. They are the ones that I usually like. I assign a service that have basically all of them, and I switch according to whichever the KI. And according to whichever task I have at hand, and it's amazing, especially for deep research. And I think the problem with AI, especially this chat services, is a bit like alcohol. It's better for 18 plus, and used with moderation, but it's not bad in itself.
Yeah, I agree.
Yeah.
I agree. I think this comes up again with the issue of ethics. That is I think that this dimension we have to face at the university everywhere. How to make the students, how to avoid the bad use of these tools, and how to detect it, because they cannot detect. I think, in my personal view, I think the large language model generation, in terms of detecting if the student itself written that or it was the AI, I think for me it's a lost battle. I could be wrong. I might be convinced against that, but I think this is a lost battle. We had to face that and then put the efforts in other areas instead of trying to discover if the student had cheated or not. I might be wrong.
Well, thank you. I fully agree with you with basically everything you said. I just wanted to tell you that there's another tool that is called SciSpace. SciSpace is amazing.
Yeah.
I mean, usually when I'm going to study a new field of new area of expertise, then I drop my initial questions there, and then it only takes information from published papers, which means it doesn't hallucinate. And then I go through each and every one of the references. Then I take it to other tools that build a huge network of literature, and then I started pinpointing the key papers, and then I download them and I add to my reference manager. But I mean, it's just to tell you that SciSpace is also amazing.
Yeah, I use that too. But it's almost a kind of personal preference, the way that I thought more friendly and the return.
Regarding the students, I myself teach biochemistry and programming, and I encourage students to use all these tools. I just tell them use them correctly, use them to learn and not to hinder you from learning.
Yeah.
And I try to convince them, like on a daily basis, that they shouldn't sabotage their own learning, and instead use the tools to benefit. To benefit from them.
But I think that the way to change that, and that's why I say that the universities have to change, is the way that the students are evaluated.
Yeah.
So written exams, and for me, doesn't make any sense at all. We had to change this. The question is how and how fast you have to do it, right?
I agree. I think written exams have their place at some subjects, not all, but we have necessity for new types of exam. For example, if you are doing like some deep math or mathematical analysis, physics, mathematics, you have to go through written exams. No way through, I think, all around that. That's my perception. But you also have to incorporate some kind of, okay, how do you chat? How do you program this on a simulation? You don't need to abandon the past. You need to go forward also.
I think this is a more deep discussion. We have to face that.
Yeah.
That technology is overwhelming us.
Technology destroys empires.
Então, agora temos a Mariana Simões. So we have Mariana from the Institute of Biophysics.
It's from the group of Pedro Pascucci.
Está desconectado. Onde é que eu coloco aqui para apresentação?
É no Google?
Não, full screen.
É no
Full screen.
Esse é o check sim.
Não,
Ainda tá, ela tá ajustando ainda. Depois ela vai conectar.
Eu
Tem.
É esse aqui, né?
Não, esse é o meu.
Okay, so Mariana.
Tá. O microfone tá
Então, mas eu acho que
Good afternoon, everybody. I'm Mariana. I'm a PhD student from Professor Pedro Pascucci's lab. Okay. And we are from the Institute of Biophysics, Carlos Chagas Filho. We are UFRJ. And in our lab, thankfully, I'm talking after Professor Pedro Torres because he's one of the professors at our lab. So we are investigating biological problems and phenomena with computational analysis. We are mainly a computational lab. So the work I'm going to be presenting is the pattern recognition of fingerprints on molecular interactions using AI. This work is done together mainly by José Anchieta, another PhD student, myself and Professor. The first thing we have to think about is the resolution of structures when we are studying proteins and macromolecule from biological systems. We have here, so we have the resolution of electron density. We can see that the higher here is the size, the 3 angstroms, 2 angstroms, and 1.2 angstroms is a unity, of distance. And when we have 3 angstroms here, we have a lower resolution, and we are not able to understand and fully comprehend the structure of a molecule. And we have advances that came, especially with cryo-EM microscopy, and we are able to have a much better resolution and to understand how the molecules they are positioned and how they can interact with each other. And the first thing you can think about is a protein and ligand inhibitors interaction. And the assessment of electron density, which is one of the, what in the structural field we access, can be done by calculations involving quantum methods, and they are essential to better comprehend the structure and the biological molecules. So here we have the electron probability, of one atom, and then the counter, so we can see how these orbitals, they occupy the space, the three-dimensional space surrounding the nucleus, and the radial wave function of atomic orbitals may reach large distances here. If an orbital, an atomic orbital, the wave function can reach large distances. It means if I have another atom next to it, if they are in a smaller distance, they can interact with each other. And then we will have the interaction and another wave function of the whole molecule. Here we have the visualization of intramolecular bonds of molecules and the hydrogen bonds between them through a method of atomic force microscopy. And then here on the right, we have the interaction between protein and then a ligand. And then we can see the electronic density in red of each one of so from the protein and then the ligand. And then we have density here in the middle of them, which is in the intermolecular space. So the main goal of our work is to build a machine learning algorithm to recognize these patterns on a database of fingerprints on molecular interactions obtained with QM calculations, quantum method calculations, of complex structures at the molecular level. The first thing to do that is to investigate the role of the intermolecular orbitals in molecular recognition through the construction of interaction intensity maps of atoms and residues, which is the image that I've shown. Here we can, so this is the assessment of the sharing of the molecular orbitals. Here we have a complex of a protein and substrate. So we have here the protein and the substrate, and we can see that the superposition of the atomic orbitals in the enzyme complexes associated with, in this case, the substrate. We have the highest occupied molecular orbital, which is in the frontier. It's involved directly in the interaction with other molecules. So we have its sharing. So this orbital is both in the ligand and in the protein. That's what's very interesting about this result. And we can see in this other representation the localization of this electronic density in the intermolecular space. And we have to think, about the wave function and the parts that became the density itself. So the sharing of protein ligand molecular orbitals suggests a role on interaction energetics, molecular recognition signaling. Another system that we've been studying is protein DNA, and then we are observing here the sharing of these orbitals. So we have here, it's represented for better visualization, the orbital in the DNA and the orbital in the protein. Here we have as a heat map the bases of the DNA against the protein residues. So that's what we are calling fingerprints, because this intensity map show the signature for a protein and how it will interact with the DNA. Okay. A second part is to build a database from the PDB data with the module of pattern recognition of fingerprints through intermolecular orbitals to train our algorithm. And then we are correlating, so we have molecular orbitals correlation to binding energy. In order for interaction of a ligand to a protein to be more stable, it has to have a lower energy. And then here we are accessing it, this interaction, this energy, with a KMM method, in which we are accessing the region of the catalytic site with the ligand, with quantum, molecular, and mechanics, and then the surrounding with molecular mechanics. So the idea is to build the algorithm capable of recognizing these interactions between ligands and enzymes, aiming to train it with experimental data. Here we are observing so we've done this with a database of 48 complexes of beta secretase in complex with different ligands. And then we can see this correlation of the electronic intermolecular density against the energy, the binding free energy, and then which gives us we are more sure about the correlation of them, and then we have that with other pathological systems. Here is an example of a protein that I've been working on, which is an enzyme of the malaria agent, Plasmodium vivax. And here on the left, we can see the density of orbitals, and the orbitals on the frontier orbitals are HOMO and LUMO, and they are located at the catalytic site. So their activity is represented as the susceptibility of the attack and between the atomic and molecular orbitals of the protein and possible inhibitors. In the end, we want to validate this algorithm based in the KM data of microorganisms enzymes with candidates of inhibitors. And we want to validate it. So we will reach different databases, and then we'll evaluate the patterns and have the result. Thank you very much. This is our group. Professor Pedro Pascucci and José Didier here. And thank you very much.
I have a brief question about the models that you use of the proteins. The question about the models that you use for inferring the interactions, right? And by the way, very nice work. Were they already solved by any of experimental method, or you used AlphaFold? And actually the main question is, what are the resolution cutoff that you normally use for that? Because I'm always afraid of the resolution of the context, right? Because you may have like some flexibility dynamics over there. So.
Yes. Okay, thank you. I didn't mention, but the cutoff was below 1 angstrom. So we wanted it to make sure that we were observing the density of the electronic density of that atom and that molecule. So that's below 1 angstrom.
Okay.
And about the structures, so they were evaluated, the test with the 48 were all experimental.
Probably
yes, data that we want to train it with the experimental, and then we were able to apply modules or new structures throughout time.
Thanks. Congratulations.
Às vezes, se for fazer grau.
Pode demorar muito não, porque
pode ficar apresentando no canal. A apresentação não, ele já tinha comentado.
Necessitar o som.
O AlphaFold 3, então, ele está conseguindo definir interação. Pensei que eu estava falando das frases.
Ele vai fazer, agora se for bom, né? Ele sabe.
É.
Mas a gente pode fazer, eu acho.
Eu acho que sim. Pois é. Porque assim, o que eu estava pensando era algum programa que a gente possa treinar com o nosso database específico. E aí pegaria todas as interações que existem no PC, dinamocode,
uma pendrive.
Um pendrive. Aqui pode colocar.
É B externo do externo.
Ah, tá.
Certo. Se funcionar, vai.
Legal.
Do externo.
Eu lembro do meu disco.
Eu passei um microfone aqui. Eu acho que ele voltou, não.
Subiu aí?
Foi.
Tinha dois.
Tinha dois.
Eu lembro que dei um para você.
Talvez precise da minha digital para seguir.
Pode ver.
Ah, não, o outro tá aqui, já vi.
Deixa eu botar o dedinho ali. Eu acho que precisa da digital para ligar.
Pode ver.
Esse daqui é de quem?
Esse foi.
Esse é?
Não,
Você
foi
vai.
não.
Ah,
Sim, tá falando.
aqui, para você não esquecer.
Obrigado. Vai querer o pointer também?
Pode fechar essas aqui?
Pode fechar tudo.
Collaborating with Bruno Motta of the Institut, but he himself is from LSSC.
So thank you for having me here. It's an honor to be here presenting this work. This is a very early stage presentation from our collaboration with Bruno Motta. He's absent due to being in another event outside the city. So the motivation for my work is about training of both large language models, computer vision models, which is usually when you follow the training of these large language models, you follow through indicators like loss, in training indicators like in training accuracy, and these have limitations because you can sometimes overfit on the training set and do not generalize, and it'd be useful to have other kinds of intrinsic indicators of quality of your neural network. And some of the researchers in the last few years have been searching for this kind of intrinsic indicators. One of these is this group, Charles Martin, Peng, and Mahoney, which have been using ideas from thermodynamics when they envision the idea of a neural network as a complex system where you can understand the neural network in which they would behave as a cascade, a kind of a fractal system, and in which the power law of the decay of the spectrum of this neural network would imbue the information necessary to indicate how well trained the network is. So the basic idea is that if you have a layer of your neural network, you would be able to, you have a weight, you decompose via a single value decomposition, you have a spectrum given by the single value matrix, and this single spectrum have basically empirically you observe this spectrum have a form of a truncated power law. And the trick is, this truncated power law has a somewhat universal behavior when you have a well-trained network. This power law spectrum have an exponent around 2, according to the authors, for well-trained networks. That's a statement that they make, and that's something that they allege that they have observed for computer vision networks, for early, like Llama, because they have example for Llama 2 and some early BERT networks. And that's something that piques our interest because it doesn't depend on neither the test nor the training sets. So something intrinsic to the network. And the argument is that it has to do with the thermodynamic behavior of the network. You can understand this as a complex system that equilibrates in a sense of a thermodynamic landscape, and you can bring to about tools like renormalization group and other tools from physics to understand this kind of system. So the idea is they propose this, they have indicators like this alpha hat, which is a kind of log pin norm of your of the weight of the eigenvalues of the matrix, which can be used as an indicator. So they get, for example, for a computer vision net of different values, and they observe this correlation between this kind of intrinsic indicator and this kind of extrinsic indicator, which is the top 1% quality of this network related to this accuracy against this intrinsic indicator, which doesn't depend on either on test or training set. You also observe this for the other ResNet, and you also observe for this absolute exponent, which you draw from the spectrum density of this GPT-like, which are not computer vision models. These are from text NLP-like models. So you have this very curious, complex system behavior appearing on machine learning systems. For our group, this is nothing new in the sense, because neurological systems have also presented this kind of complex behavior. For example, you have self-organized criticality in cortical networks. This is something that is known for biological networks. This is also known for oscillations in activations on in neurological networks. And in particular for Bruno Motta's group, he has this publication with Susanne Herculano-Ruzel, where he demonstrated the appearance of these power law behaviors connecting the geometry of cortical folding in mammals, both humans and other mammali mammals, in which you have this cortical thickness, cortical surface, exposed cortical fold surface. You have different kinds of fractals depending on which, whether you have humans, elephants, cetacea, but you have this kind of universal power law that connects all of them, who are connected to one kind of universal fractal. So this kind of thermodynamic-like behavior of complex system is nothing new to this kind of group. So that's why I approach him with the idea of bringing this kind of thermodynamic approach to neural networks. So what I propose him, bringing this kind of TechMech approach from biological networks to artificial neural networks. But since these neural networks from computer vision and natural language processing are somewhat difficult to deal with, because you have the network and you have the data sets. One thing that we wanted to do is to control a bit better. So the idea was, why we don't control also the data set by using a physics-informed neural network? Because you have a physical problem with an exact solution we can control. And we can have another fixed point, and can have another way to check if the network is good or not, besides in a sense, the top 1% error. You can compute the loss, but you can put the residue in the other idea. So the idea of physics-informed neural network starts with Raisi, Perdiadakis, and Karniadakis, where they introduced the idea of instead of using a grid to approximate the solution of differential equations, you can use a network to approximate the solution, and you treat the problem of solving a differential equation as a problem of training a neural network, and you put the residue of your differential equation as a loss term, and your boundary conditions are another part of your loss term, and you feed that into your training library. So you can nowadays use a very optimized libraries to do that, and we can do all the way to machine precision, so you can be very sure of the quality of your solution. That's why you can use this as a control group, in a sense. Since we can do very close to analytic solutions, you can use the network that were trained for these problems as a control group and look for what the weights of these networks look like from this thermodynamics perspective. And this can be done for very complex systems. For example, this is a very nonlinear dispersive EDE with very complex chaotic behavior. And if you can see, for example, the loss behaves very strangely, and still it's able to converge fairly well to the exact solution. It's how it can do that, I don't know. But that's something that we aim to be able to answer maybe in a year or two. So that's an idea. So back to our proposal. So go back to this basic framework, but with an anchor. The anchor being, we know our physical system. We are able to establish the exact solution because we know either the analytic or a reference point. And we can compute for those systems, the neural network that solve the systems very well, and we can analyze through this framework to know whether or not this framework really makes sense. Might not, but it would be interesting to know. Very well. Thank you. Any questions?
Question? You go. Adriano? We're closing to the end. We have just Adriano and Claudio. And then we'll have a musical surprise.
A musical surprise.
Yes.
Okay. Eu não sei.
Não, agora não. Tem que ser no final do programa.
Yes.
Não, eu queria o pointer.
Nossa, Pedro, você é indispensável hoje. Hein?
Pedro é sempre convidado.
Sempre. Vou colocar.
Eu vou conhecer.
Cara, eu teria conseguido fazer o tempo. Acima do que eu consegui.
Vamos falar.
So now is Adriano Cortes. So Adriano is a colleague from the Institut Mathematica, but makes the bridge with also copy. Is of the group of our team. And he has also international experience, like he was in England.
Yeah, in Kausky, Saudi Arabia.
Saudi Arabia. And so he's going to talk about his research on systematic.
Yes. Okay, good afternoon. Thank you very much for the invitation and spot here to talk about. Okay, this actually it's a new project. It's already submitted. It's a collaboration with different institutions. Okay, okay, it's here. UFRJ, Fiocruz, also NCC and UFI. So UFI is the people from the wet lab that does some experiments. Okay, and this is CapGen LM. So I will explain what this CapGen is, and the type will explore the genomic language of multi-drug resistant bacteria. Okay, so I'm happy to do the last one. So since a lot of people, and also Pedro, talked a lot about what I'm going to do, we have a good intersection, but a different approach. So this will be nice. Actually, I have a lot to talk with later on. Okay, so okay, I'm applied math and computer science. Okay, so people from biology is from UFI, also Marcelo Trindade in NCC that work with metabolic networks. Okay, so this is it. Start with a collaboration with my colleague Fabrício Alves from Fiocruz. We used to work in gene regulation networks for
GBM, the glioblastoma multiforme cancer. So we have some studies, but also Fabrício has, he's actually the coordinator of CAPGEN, and I go to what CAPGEN is actually is it's the network. It's related to the Brazilian genomic surveillance network related to multi-resistant drug bacteria. And we already we were already doing some machine learning work, but then I realized that okay, the first part of our network was related to an embedding, okay, to an encoder, and then we have the rest of the network that was a classifier. Actually, we are doing it now actually, trying to build a classifier for Klebsiella pneumoniae and related to the antibiotics that is meropenem. Okay, so we are trying to see if the bacteria is resistant or not. So based on the genome of the bacteria. Okay, so okay. Pedro already brought this information here. According to the World Health Organization, by this year here, superbacteria will kill a lot of people. Okay, the current microbial resistant surveillance mutations. Most methods from bioinformatics nowadays is based on homology. They somehow miss some complex resistance patterns. They lack this contextual understanding. That is something that we learned from LLMs that other than do some reasoning, and this is related to what they learn somehow and how to encode the semantics that is inside this language. So in current, they are heavily, the bioinformatics approach nowadays is heavily based on sequence similarities. Okay, so what is our science? Context-dependent resistant mechanisms, predicting phenotype outcome from genomic data, and extracting meaningful pattern from vast genomic data set. So we just need for approach to go beyond sequence matching to interpret the genomic language of bacteria. Okay, so this is what our initial thought and our premise. Let's say, just as ChatGPT understands natural language patterns, genomic language models can decode genomic grammar bacteria. So this is start also with what people was talking about deep research. So this exists. So I was started, and then a bunch of words can read. Okay, and then I realized that okay, I already heard about AlphaFold for sure, but I did not realize that's actually what AlphaFold is, a generative model. But to do a generative model, first they is doing encoder. So you have to put this information in a semantic way in a latent space. So there's why do not do this with genomes? Okay, so for sure people already was doing this a lot of work. Also, I will show that Facebook now Meta already has a large language model trained with a huge database of eukaryote genoma and prokaryote genoma, a lot of things. We have a lot of language about that. So why not use transformers and pictures to biological sequence? So people already done this. So you can see here you have DNA bird, you have nucleotide transformers, you have this one for Meta that is based on amino acids. This one are based on nucleotides. And why not? Okay, we know that nowadays ChatGPT is trained in a huge corpora of every knowledge the humans. Okay, so it's the new Alexandria library.
but but at the very beginning, we knew that if you ask some related to medical stuff or something related to laws, maybe he will not perform that very well because it's not very specialized in this kind of language. So the idea was, okay, why not take one of those languages and try to specialize it to what we have and what we are interested. That they are bacteria from the escape group. That's Klebsiella, and Cetobacteria, and Klebsiella pneumoniae, and the one, sorry, I forgot the Pseudomonas aeruginosa. Yeah, thank you, Philip. Yes. Okay, so that's it. So what we envisioned in that approach was that we could have some contextual understanding of the genomic elements. So because we know that's something that transformers brought into the NLP area, that's this ability to somehow see very far from the language and the context that we have, and not only what we have been doing for with LSTM and all those things. So this is actually we already have some words related to this thing here. Not my word, actually, the literature or documents. Okay, the ability of language models of this type to recognize divergence sequence, some superior detection to subtle mutation patterns. Okay, so this is what we envision as the key advantage. And for sure, our premise was this one. This approach has shown remarkable success in protein structure. So a lot of people here spoke about alpha folding. So I guess we already have a proof of concept that we can use language models to nucleotides and amino acids. So we already have it. So this was our premise. Okay. So and where our data will come from? Okay. So this project is that was originated, as I mentioned. It's part of this the Brazilian multi-drug resistance surveillance network that is hosted by SUS, MPK, and Fiocruz. Okay, and this has a national geographic coverage, more or less. I guess it's 18 state labs. So every multi-resistant bacteria that is detected in a hospital, okay, it is sent to the lab same, okay, the state reference laboratories to be sequenced, okay, and they use the CABGen infrastructure, all the bioinformatics pipeline, and then also storage this genome. So we have all the infrastructure in terms of data to specialize this genomic language model. So that is something that we found is very interesting. Okay, we can specialize this language to try to understand and then talk about our bacteria, Brazilian bacteria. Okay, so that's something we know that's. And what is CABGen? CABGen, it's a platform you can access here now, cabgen.fiocruz.org. So you can go there and do apply for using the infrastructure to see the data that is there. So it's a unified bioinformatics platform from the Brazilian health system. Okay, it has let's say a classic. Okay, all these tools are well developed, are very good, but they follow the traditional bioinformatics pipeline. So, but for sure, we are going to use them and be on top of them. But we are planning to go further. Okay, that's the point. So, and then, as I mentioned, CABGen already has all the operational and processing genomic data. So. From this network, the Brazilian surveillance network, multi-drug resistance. Okay,
So just a bit of technical stuff that what we put on the project. So you have to use some fine tuning. Okay, so and for sure now LoRa is the most used fine tuning technique. That is, you don't have to use a lot of parameters. Okay, so just using low rank matrices to update the weighting of the neural network. So it's very efficient. So nowadays it's very good. And also this is very important because that's the point. When LLM like ChatGPT hallucinates, we know that's natural language. So you look at it, like, oh, this is crazy. This is strange. What's going on here? So that has no meaning at all. So, but how they have this in the genomic language? Since we do not speak, let's say A, C, T, G. You're not allowed to speak like this. So we try to put in the submission in the project a lot of stuff. Also, I have a colleague from Harvard that is doing his research. There's a postdoc. His area is about this interpretability. So I could talk with him and put him in this submission to have this part of an advanced interpretability. So because we really have to take care about of the interpretability and reliability of those languages. So this, I guess, is the most challenging thing of this approach that we are trying to pursue. So we try to put a lot of effort in this part here. Okay, and this is what we expect. Okay, this is for now. We have, let's say, if I could say, we are in TRL2. Let's say the maturity. We are in maturity 2 in terms of technology because we know that the literature already documents some successful applications. So, but we have a lot of steps to go. We hope to. We also plan to put this in because the CAPG is also a platform, a web platform. You can go there, you can do some analysis there, you can do something there. So we plan to enhance this analysis. And okay, just give one example that I guess is here. Let's say enhanced outbreak detection. So being after having an encoder that we have this, we put the genome of the sequence bacteria that was detected, let's say, in the north of Brazil and some in the south of Brazil. So we can somehow put this in the latent space, the latent space of this semantic language, let's say, and then try to cluster those things and see, okay, even though they are the sequence is not very, very close in terms of sequence matching, but they are very close in terms of latent space. So, okay, it's bacteria in the south and the north. Okay, let's do some investigations to see if people travel from south to the patient that was detected with the bacteria was detected in the south. He traveled to north, or the north, or the patient from north traveled to south. How does those geographically separate places have some bacteria that somehow was clustered together in the latent space? So I guess this approach can have very powerful tools, can try to answer interesting questions, but also has a lot of challenges. So that's what we plan. Okay, to have a superior prediction of antimicrobial resistance to these things. Okay, so this is my last slide. I guess I passed from the 10 minutes, sorry about that. But additional bacteria and also antibiotics that we are testing.
because each of bacteria of this escape group has a different mechanism of resistance. Some are some active mechanisms, like pumping. Those are beta lactase. Yeah, it's lots of for different biology. Don't know the terms in English. So. And also, so the advanced interpretability methods, maybe cross-cultural surveillance integration, who knows, so in a project together with friends. Okay, and for sure expanding to other genomic applications would be very nice. Okay, so thank you very much for your time.
Thank you, Adriano. Question?
Specialist, okay. The specialist will talk with me.
Yeah.
I'm computer science, okay, and applied math.
So
I have biology on my background.
you have we have heard about protein language models too, right? PLMs. They use that for
Protein language. Oh,
yeah, protein language models. We have heard that from people from Facebook, people from AlphaFold. They actually use protein language models, and they use them to make predictions. So and use generative models. So what they do is they learn from proteins that exist, and they generate proteins that do not, or maybe structures that do not exist yet. My question about your project is, what would be, well, the input? I think the input is clear. I think the input would be the genomes of the
genome. Yes.
Yeah. But what do you expect as an output? Because as I understood in the end, you were talking about clustering and understanding the relationship between different genomes.
Actually, when we say, okay, when you go to the attention is all you need, the paper that popularized the attention mechanism, you see there are two different stuff. That is the encoder and then the decoder, and then the decoder you have some, then you can put another network. Okay, so right now we are thinking more in terms of BERT, that is the encoder. Say just put the language in a semantic way, let's say, or in a polysemantic. It's difficult to deconvolution the semantics and how that's why interpretability is needed. But we are thinking right now in terms of latent spacing. So put this language in the latent space, so we can on the latent space do some specialized tasks. So that's what I say. I'm not planning right now to do some generative model. Let's say we are planning to just put in the latent space in a semantic way, as I'm saying, and then do another machine learning architecture. Let's say actually maybe the classic one, not even neural network, can be like another one, like SVM or.
Are you looking at the sequence level? Are you looking at the sequence level? The proteins, I mean, the genomes. Are you looking at the sequence level, A, T, C, G, or are you looking more like a more, high level, like identifier of genes? Because that could also be interesting. Instead of looking at the individual bases in the genome, maybe you could look more like to the structure of the genome, and the gene names, for example, could be useful.
Yeah, yes, I see that it can be. Actually, there are different things we can try in this field, let's say. Right now, what we are doing, we are just using the open reading frames.
Okay.
So we are doing a pre-processing of the genome, and can just take the open reading frames that we are supposed to be where the proteins can be encoded, and the resistance maybe there somehow in the active So, and yeah, sure, we can try to pick some motifs that we already know from the literature that the bacteria family can have. Okay, that's the point. I'm not a biologist. Okay, what I know about biology is very, very restricted. So that's why I need to talk a lot with people from biology into, because this is a multidisciplinary project. So I cannot enter too deep in the biology, and I guess the people that from the biology to go too deep in the AI part, I guess it's difficult. So that's why you have to tune our talk and then go. So our first publication with Fabrizio took like 2 years to mature the language, the meetings, everything. So that's okay. Thank you.
Thank you. Thank you. So now, thank you. So now we have Robin Cherry. If I do. Yes. There are many names in just. So he's from the house. So it is a challenging opportunity.
Thank you very much. My name is Claudio Micelli. Actually, Claudio Micelli Farias. So I'm also from here, from NC and from PESC as a young, and I'm talking about time gen AI. I promise to be fast. Is it better? Okay, perfect. And my work is for Internet of Things in smart cities. I work mostly on this for the last 20 years almost. So I'm working in this scenario where I can collect data from all over the world, and what do I do with this whole lot of data? And this is quite important when you think about Industry 5.0, industry of mass customization, where IoT is one of the pillars. So if I'm gathering this data, and I want this data to customize your experience, or as I say, if you are watching nowadays Black Mirror, we can do everything on that right now. There's nothing that in that series is just not a stereotype that you can't do. So the idea is that like now those machines can think, and this is leveraged by around 100 billion IoT devices in the world nowadays. This is not from me, it's from Siemens, the project that I have done with Professor Daniel Sadok that was here before. And in this scenario, we arise what my most recent research goes is time machine learning. How do I make deep learning into 16K of RAM? How does I make that work? And this is not something that only I'm looking for, but a lot of people. And this is a very interesting device. It's called Arduino Nano 33 BLE Sense. It's a sensor that costs $10 and has a machine learning processor, our Cortex M0 processor. And this is SI. It's a sensor destined to computer vision. So those devices are done already in production. We use this a lot. This is a book from O'Reilly, already 4 years old, if I'm not mistaken. And how do we do this? So we do the exactly same process that we do in the traditional deep learning, but we have to now try to distillate, concise, and compress those models. And this is very interesting, but it is not new, because I can just quantize my models and try to summarize. But since I have 100 million devices or thousands of devices, those distributed answers are usually better. So, okay, instead of one big machine, I have a cell phone with 8 processors. Everyone here has one. And those guys already use AI for everything. If I could cover a city, I can have real-time assessment of any situation. And then I can do better, because now instead of having a whole model inside of a sensor, inside of a cell phone, I can have parts of those models inside of sensors or cell phones and distribute it. So if this room, the temperature is like 20 degrees, 25 degrees, I don't have to understand the whole range of temperature. I don't need all the parameters. So I can do student models that can be traced inside of embedded devices. Again, I have 100 million, 100 billion devices. And then I can even cut it out. I can prune all the connections that make no sense. And then a big model can be cut into a smaller device. But this is no nothing new. We've done already. There are several papers on this. But the interesting thing is that those same things can work in LLMs. Now imagine that your sensor is not actually sensing the world, but predicting the next behavior, correcting elements of what you propose. And this is interesting because in many, many areas, LLMs are better than traditional LTSM models, than traditional regression models actually too. So this is not something that we are working on, but actually Meta is working on this too. So this is from Yannick Kuhn, I think it was last year. It's his paper on mobile LLMs. But we want to go further. We want to use what is now called the hive mind. Use all these sensors as a perception layer of reality. How can those sensors understand the reality and predict your next behavior on real time, exchange with the environment? Remember those, there is a joke on the AI community. There's an old lady crossing the street, and now she doesn't see the red lights, and the car is going to hit her. What does the car do? Now the car doesn't do anything. The whole street does things. And this solves the problem. And as we can see this, the idea is like those generative models inside of sensors can be seen as a perceptive organ of an environment that we use to perceive as something not sentient. So these 100 billion devices that are nowadays with a perspective of 1.5 trillion devices up to 2035. But those 1.5 trillion devices now have all capabilities of learning, understanding, and predicting on real time. And exchange this knowledge, creating a new way of how to interact. So a lot of things that we have been saying about web semantics in the early 2000s, nowadays AI is taking over. 10 years ago, we were seeing things on blockchain, but if you could mixture AI and blockchain inside of a same semantic layer to create rules and to predict next steps, this is something very powerful. And what we are doing right now in my lab is developing new hardware accelerators for AI. And how can these things do not deplete energy on very, very fast? So we are working with Brain AI a lot right now, and especially on data fusion, how data fusion methods can be seen to leverage use of filters can give us those LLMs faster results. And I'm organizing a conference called Fusion on July. It's the biggest conference on big data topic, trying new types of neural networks inside of those generative models, like weightless neural networks and hyperdimensional computing. Work a lot on few-shot learning. How can I predict when I have almost no knowledge about this environment? And work about transfer learning. Transfer learning is already established, but not in 100 billion scale. So how can I do and work with distributed systems on large scale in real time? Or how can I do and interestingly, a lot of those things goes to game theory. And this leads us what nowadays we call one very interesting step on these generative AI models for IoT is federated learning. One way to exchange information is simply not exchanging information. It's exchanging parameters, because those parameters is how we understand the world. So these parameters distributed among sensors, instead of representing one single reality, those parameters together can build new reality. So each sensor can understand parts of this reality and summarize this. This is what we call aggregation methods. So instead of aggregation data, we are aggregating semantics. This is very, very powerful. Is where we are working right now, especially with CON networks, Kogamura-Garland networks. Very, very interesting. And the results are not good until this point, but that's something we're working on right now. I have some works on ICML and NeurIPS right now to try this. And some of the core challenges that we are working on this is most of the works in literature are very homogeneous. And when you have this reality of 100 billion or large scales, devices are heterogeneous by nature. So we are talking about differences in processors. So even the words of the computers are very different. Communication is still expensive, even if you quantize, compress, or even change. There's something that 10 years ago, I was in an IoT conference, they said to me, Cloud, in 10 years, bandwidth, memory, and processing power will not be limited. And 10 years later, yes, they are not the same from 10 years ago, but they're still limited compared to what we need right now. So there's always this run for the gold. And stragglers are actually one of the biggest problems because since those devices are heterogeneous, they don't understand the same thing at the same time. So how can we wait for them to understand everything so we lose the conference? So the fastest has to give this approach, but this is a problem in distributed systems. If just a small part take the decision, how can we trust this decision? So this is complicated, it's still working on. Actually, we use subjective logic to try to work on this, and it's good results, but not what I expect right now. So we are working right now in advancing neural architectures. We are working on time machine learning specific habits. And so this brings us new opportunities. We have a lot of interaction with universities in Europe, especially with Udine and Calabria, and some opportunities also with China. A lot of my partnerships are with China, so this is interesting. My Chinese is awful, so. And that's it. I try to be as fast as possible. Thank you very much.
Thank you for the presentation. I have two questions. First, on the stragglers, the first thing that came to mind was latency. Probably that would be by far the worst of your problems, because you have a network of stuff with heterogeneous networks, with horrible networks. You are not working on a cluster with InfiniBand, all nice stuff. You are working on cell phones. That's not workable. But one thing that came to mind on stragglers, you might be interested in the literature for exascale integration, exascale simulation software, because they have these problems porting codes, because they had stragglers even on high-speed networks on these big computers, and they developed basically soft lock integrate communication protocols to be able to deal with that. The second thing, the energy constraints. Most of these companies are moving towards monolithic solutions rather than the best example for this is Cerebras, which basically built like one wafer scale solution. For me, like the obvious solution. Why go the other way? Because you're cutting on wires, you're cutting on transmission, you're cutting on communication, you're putting everything on the same silicon. That's obviously better.
Actually, this is a problem of cost. So,
yeah, but that's much much less expensive per core, per byte, yeah, per everything.
But
it's not per unit of rack is less.
Because the idea is not to build something; it's to reuse things that are already there. So that's the main purpose of using those microgenerative. Because since we already have cell phones, you already have, like in your house, smart toothbrushes or smart.
But it's a scrappy solution, right?
Yeah, it is a scrappy solution, and that's very, very different in the sense that because those computers are not close to the monitored phenomenon. So they have different understanding layers. For instance, one case, all these things are very interesting to take decisions. And for instance, it's like
weather monitoring. I understand. Like
okay.
you spread sensors about. Yeah. Yeah, close. Yeah, sure.
No, we can discuss.
Yeah, sure.
But the idea is like, so, since some decisions cannot be taken afar from the phenomenon, so those decisions we can decide. So it becomes a scheduling problem.
Sure.
Where to take the decisions and to scrap those resources that are already there. For instance, it's one of the the style machine learning is a trend where most of the global south can still have a lot of impact because those resources are already here and you can use it. And this change then can expand. So those big clusters, those big brains, they are part of the solution, but then not only they will enclose a lot of the processing and a lot of needs, but those we have to decide when I'm going to use those.
Sure.
Okay, this is the what we can discuss. There are a lot of small. Thank you.
So I think we have to skip the music from just to Paulo Mann, that is from the Institute of Computer Science here. He's the youngest professor, the latest. So you will become
Thank you very much. So today I'm going to present you a little bit about my recent research and a little bit about the past and the beginning of the research as well. And they are all related to social media in general. But most important, what's social media analytics? So basically, we want to study the dynamics of social media, how people disseminate information online, and how people spread misinformation. And more recently, we're more concerned about the mental health of people that are using social media, etc., etc. So social media provides us a very important feed of information that we can use and rely to extract several informations and several insights from it. And in my group, we rely basically to find those kinds of information that I will talk more later. But we rely on this kind of areas, so basically artificial intelligence more broadly, and large language models as well, of course, and sentiment and topic classification and multimodal analysis, because when you're dealing with social media data, basically we're relying on images, audio, text, and videos, so we are dealing with a very different kinds of media online, so we must deal with all of that information. So basically we use that kind of areas of research, but my research group, we are most focused on mental health research on the first area that we are studying more on social media. So we basically want to use and rely the social media data and understand how we can screen and pick up who are depressed and who are not depressed that use social media. So we want to understand if we can use social media data to find those people that are suffering from major depressive disorder, and we want to basically try to help them, guide them to find therapeutic interventions. The other area of research that we are also concerned is public relations. So we can find, for example, the general sentiments toward a company, and we want to understand how people engage with this company, whether this engagement is positive or negative, or whether people are talking about sports when are talking about a specific company, or what kinds of topics they are talking when they mention a specific company. So this is the area where we want to try to manage the image of a company within their channels of communications. In the third and final area, we are dealing with finance, where we want to understand how people share news in social media, and are those news related to material facts, whether people are saying something good about an asset or something bad about an asset, and how this is spreading in the social media, and how this could impact the value of an asset. And so this is very broadly and in general, this area of research we are doing here, but I will focus more on the mental disorders aspects. That is the research we had more focused on my group. So there are many mental disorders, and for example, here we have depression disorders, anxiety disorders, and bipolar, and we have many others. But in our group, we are specifically focusing on depression, where we are trying to screen for depressed individuals that use social media, and how we can find the relevant topics that they are talking in social media, and whether we can differentiate what kinds of topics they are saying in social media, like from people that don't have major depressive disorder to people that have major depressive disorder. So basically, we can use three ways to intervene in this process. We can use AI to help the diagnosis in a way that diagnosis is only conducted by a psychiatrist, but we can try to help to support the diagnosis process. But we can also use the AI to help in the treatment process, and we can see people using and relying on chatbots to get therapeutic treatment. So, for example, I have seen a post on X that a female user and AI researcher was trying to get help from ChatGPT after her cat died. So, those are the ways people are trying to use it, but there are problems and pros and cons, of course, and I'm not here to discuss that, of course, but there's something that we must discuss as well. So we rely on social media to do that. So here is basically the general overview of my past research and my current research as well. And I started on the International Conference on Web and Social Media in 2020. So we all started there, where we were preoccupied with whether we could verify if students from the Federal Fluminense University, whether we can use the data they create on social media to try to help them and screen the depressed individuals among the non-depressed individuals. And we all started there. And more recently, we are currently trying to understand the topics of what people are saying on social media. So for example, if I'm using Reddit, I'm using X, I'm using Instagram. So what kinds of topics, if I have depression, what kinds of topics I'm saying there, and what kinds of topics people that don't have depression are saying on the social media as well. So we basically trying to extract latent themes where we can understand what people are broadly saying about anything in the network. And we're also trying to generate synthetic data for like further improving the performance of those models. And we are trying to improve that based on the existing data we have that we have collected. And we are trying to improve those models by relying on this existing data to generate synthetic data that we also know that have many issues associated with that. But we can see that the model, the resulting model often is much better in terms of accuracy to detect the depressed individuals. So that's basically it, and try to be as concise as possible. And that's it. Thank you.
For persisting this marathon. We had to announce to the last documents online, but it was about music because we also had an intersection between the Institute of Mathematics and the graduate school in music, which is a MUSMAT. And so they basically use Hugo Tremonte, that is our colleague, they basically use sort of machine learning to try to have a DNA of the composer and to be able to compose new music in the style of some composers. So they analyze partitions and that the data, huge database of musicians and especially popular music in Brazil, to different mathematical tools for this. And so this is fun. He couldn't be here and it was online. So thank you all, and thank you for the people for this actually Pedro. I had a comment to your comment, I agree, about the modeling. question. Thank you so much for also Hugo from LHSC, is another, all very interesting comments. Thank you to everyone. And see, I hope we provide all the emails to you. And if you could provide the presentation so we can share with them and they can share with you. Yes, yes. Thank you very much. And thank you for the last talk as well.